Hello, fellow biologists. This is Dr. Tarun again, and we are now in section 11.3 of chapter 11. Here we are going to discuss meiotic cell division. Your reading assignment for this section is found in pages 231 through 241 of your textbook. So, the general learning objective of 11.3 is for you to be able to describe the general process and explain the purpose of meiotic cell divisions or meiotic cell divisions. There are nine sub objectives here, and it's worth going through them so that, again, because this is a large material, the learning objective helps you to focus on what you need to uh, learn and pick up as you listen through the video lecture. So 11.3a is to establish the roles meiosis and fertilization play in increasing genetic diversity. So these processes go hand in hand in the life cycle of any eukaryotic organism. B is to determine the effect of meiosis on chromosome number. And three, to describe the transition of ploidy that occurs during fertilization. The word ploidy simply means the number of chromosome sets that a cell or an organism carry. So it could be a haploid or it could be diploid or even higher ploidy levels. Fourth, predict the effects on chromosome number in each stage of meiosis. So we will be focusing mainly on understanding the dynamics and mechanics of the way chromosomes gets distributed in each stage of meiosis. So we need to focus on the behavior of the chromosome in this section. So the next sub-objective would be to differentiate between the events that occur in meiosis 1 and mitosis. Next, to explain how genetic diversity is generated through the alignment of homologs and crossing over during meiosis. So, an important concept that you need to understand here is again the concept of homologous chromosomes and what is the significance of crossing over during the meiotic cell division. Then compare and contrast the events occurring in meiosis 1, meiosis 2, and mitosis. And then provide an argument supporting the hypothesis that meiosis evolved from mitosis. And finally, you should be able to relate differences in the division of the cytoplasm during meiosis to the production of eggs and sperm. By the end of this section, please also spend time to do the self-assessment question to assess your understanding. So there are three questions here. One, in a human cell at the end of Prophase I, how many chromatids, centromeres, and bivalents are present? So you should be able to understand what these three terms mean: chromatids, centromeres, and bivalents. Centromeres here is misspelled. There should be an E after R here, centromeres. 9. What are three ways in which meiosis 1 differs from mitosis? And lastly, what two processes during meiosis results in genetically unique daughter cells? Let's have a quick overview of meiotic cell division before we go into details. First, meiotic cell division results in 4 daughter cells. So this is in contrast with one cycle of mitosis where you end up with 2 daughter cells. And each daughter cell contains half the number of chromosomes as the parent and are called haploids. Okay. Again, this is in contrast with mitotic cells where you still end up with the same number of chromosomes and the daughter cells still remain diploids in the case of obviously diploid cells. If the diploid cells start starts mitosis, at the end, it will still be diploid. For meiosis, you start with a diploid cell, but you end up with daughter cells that are haploids. Now, each daughter cell is genetically unique. This is another important difference with mitosis, because in mitosis, if you remember, the daughter cells are essentially clones of the parental cell. And we will see here how this happens, how come the daughter cells become genetically unique. Now, each daughter cell can become sex cells. This is important, or gametes that can combine with other sex cells of the opposite sex during fertilization in order to form new diploid individuals. So the core concept here is that meiotic cell division is essential for sexual reproduction, the production of offspring that combine genetic material from two parents, which explains the fact that if you look at your brother or sister, although you have the same parents, you still have some dissimilarities, although there are also some shared traits. And this comes about as a result of processes in meiosis and fertilization. Now, it's important to recognize at the start that meiotic cell division includes two rounds of nuclear division. Okay, meiotic cell division consists of two cell divisions called meiosis I and meiosis II. But these two cell divisions are essentially describing what happens in the nucleus. Okay, so there are two main important things that I highlighted here in blue that you should remember to be the highlight or main point of each of these division or general stage of meiotic cell division. During meiosis I, homologous chromosomes recombine. That is an important word to understand, which means to exchange parts and therefore to exchange genes and separate. But during meiosis II, instead of homologous chromosomes, it is the sister chromatids that separate. Now, before prophase I of meiosis I, DNA replication also takes place in the same manner that is discussed previously during mitotic DNA synthesis. So we don't have to revisit cell cycle here, but try to remember that whatever happens during the interphase stage in mitosis do also happen in meiotic, intervening meiotic cell division events. So let's now look at each of the stages of meiotic cell division, and we look at the first general division, meiosis I. And meiosis I is also divided into substages, and the first of it is called prophase I. There's a lot of things that happens in prophase I. So first is that chromosomes first become visible as thin threads. Again, they are, in this particular case, they are called chromatins, but they continually progress to condense themselves to become more visible. So again, just like in mitosis, in animals, there would be centrosome, which are the microtubule organizing center. You will still see the nuclear envelope at the start of prophase I, and then, of course, the condensing chromosomes. Now, please take note that at the beginning of prophase I, we assume that the genetic material in each of the chromosome has already replicated during S phase, the previous S phase in the interphase stage in preparation for meiosis. Okay, and then in 2, you can see that the homologous chromosomes become more condensed so that you start to see them under the microscope, and then they undergo a very important process called synapsis. And you need to remember this term. synapses. This happens only in meiosis at prophase I and not in mitosis. And what happens here is that homologous chromosomes starts to pair up, seek each other and start to pair up. And meanwhile, microtubules are beginning to get propagated from the centrosome, and the centrosome starts to migrate towards the opposite pole. And then in the third stage of prophase I, when synapsis is complete, or the pairing of the homologous chromosome, they now become what we call as bivalent pairs. And bivalent pairs means that the chromosome has come, homologous chromosome have come in close contact with each other, so that each of their corresponding genes, remember homologous chromosomes have similar set of genes. Not only are they of similar set, but their order in the chromosomes are identical. Okay, although the gene in each of the corresponding point along the chromosomes may not have the same version of the gene, but it is still the same gene. Maybe it's a gene for eye color, but the flavor of that, maybe one might be a brown, codes for brown, and the other for blue. But nonetheless, they are neatly paired, aligned with each other in the bivalent chromosome. So here, it is now important to recognize that after synapsis, bivalent chromosomes are formed, okay? And the bivalent chromosomes are made up of the paternal homologue and the maternal homologue. And the reason is because these cells that we are dealing with here are diploid cells, okay? And they are sometimes in genetics, as you will later on encounter in your future chapters, is represented by the symbol 2n, where n represents a set, one set of chromosomes, okay? So these diploid cells come about as a result of the your one set of your chromosome comes from your father, and one comes from your mother. So that's why all of your chromosomes have corresponding homologs. And so at step 3 of prophase I, they all form what we call as bivalence. And this is very significant in terms of ensuring the proper distribution of these homologs to daughter cells, as we will see shortly. So why do these homologous chromosomes have to synapse or meet together and align themselves neatly? And so this is the reason, because an important event happens called crossing over. And this process here of crossing over is the basis of the uniqueness of the products of each daughter cells that are produced. Okay, so remember in when chromosomes synapse, or when there is synapsis or pairing of chromosomes between paternal homologue and the maternal homologue over here, okay, what you have would then be, because notice that they are double-stranded arms, right? Now, remember that each arm corresponds to a double helix. And so the fact that they are double-stranded, if you recall, is because they have duplicated themselves or replicated themselves in S phase. So you have one single centromere holding two strands or chromatids that are identical to each other. So the sister chromatids, okay, initially are identical, okay, but the non-sister chromatids are not identical to each other with respect to the flavor of each of the genes that they carry, because one comes from the mother and one comes from the father, okay? Now, when the close synapsis is fully formed, it results in what we call as a bivalent or paired homologous chromosomes, which then starts to um, come in contact with each other. In other words, the non-sister chromatids will start to establish physical contacts called chiasma, or the plural for this is called chiasmata. And these are the visible points of contacts between non-sister chromatids. So I emphasize here, non-sister chromatids during crossing over. And because non-sister chromatids are not identical to each other, what happens then during crossing over is that they exchange segments so that you end up with chromosomes that have shuffled genes, okay, which therefore generates a new combination of gene flavors, or what we call later on in as you will encounter in genetics, alleles. Okay, alleles are simply different versions of the same gene. So as I said, it is like the example of the gene that encodes eye color. Although that is a gene for eye color, it may actually have different flavors or different alleles for eye color trait or eye color genes. Okay, so this is what happens during recombination, where you shuffle different flavors of those genes depending on what the paternal and the maternal homologs carry. So in Prophase I, and towards the latter stage, therefore, having seen what chiasmata causes or produces, the chromosomes will continue to shorten and thicken, and the chiasmata between, which is the point where crossing over or exchange of segments are happening, becomes even more apparent under the chromosome, not under the chromosome, excuse me, under the microscope. And finally, the nuclear envelope begins to break down. And once the envelope begins to break down, that marks the end of Prophase I. The next stage of meiosis I, is prometaphase I, which is closely followed by metaphase I. I would like to add a few more points with regards to the crossing over event. This process of crossing over, which happens during prophase I, is a random process that results in completely unique chromosomes when meiosis is complete. So please take note of that. And during this process of crossing over, there is no genes lost, or no nucleotides even are gained or lost in this process. And the other important point to consider is that, as we will now see in prometaphase I, this crossing over, the formation of chiasma or chiasmata in plural, also helps hold together the bivalents for metaphase I as they move towards the equator or the middle part of the cell. So in prometaphase I, the spindles now attach to the kinetochores on the chromosomes. Okay, so remember, the kinetochores are those complex of proteins that are found in centromeres, where microtubules from the spindle are attached. And once that happens, that physical connection between microtubules and centromeres in the individual chromosomes of the bivalents happen, then metaphase I ensues. And here the bivalents, or the homologous pairs, they line up in the center of the cell, like so. Okay, and these bivalents actually line up and are oriented randomly with respect to each other. So here, what we see are two bivalents, okay, and their position in the central parts, okay, of the cell, how they position each other has does not influence the positioning of the other pair. So these are all independent or random. But notice that because the bivalents are still connected by synapse, they are always joined together. So this is absolutely different from what happens with chromosomes in mitosis. In mitosis, individual chromosomes don't pair with their homologous partners. So they go to the equatorial plate in metaphase independently of each other. But in metaphase 1, this is very important, and I will emphasize it again. It is the bivalence, the homologous pair partner from mom and dad goes to the middle together because they are synapsed, okay, and because and crossing over has taken place, and so this is very important because this underlies a principle in Mendelian genetics that you will encounter later on in chapter 15. In fact, I put the page here, okay, called independent or random assortment or orientations of bivalence. So to repeat, the way these bivalents are oriented, whether it is blue on this side and the lighter blue on that side, does not influence the way the orientation of the other pair is in the center of the cell. Whether this one blue of the small chromosome will orient themselves on this side, or and the light blue on the other side, or perhaps the blue on this side and the light blue on the other side. So there is a 50-50 chance that this can flip, and that 50% chance of this flipping is not influenced by the orientation of the first bivalent. So this is called random assortment and orientation. And I hope that you pay attention to this behavior, because this will explain the laws of the segregation and transmission of genes according to Mendelian principles in chapter 15. So the next stages of meiosis 1 is anaphase 1, and quickly followed by telophase 1. And if you notice that the alignment of the bivalents in metaphase 1 does something remarkable, and that is it ensures that the two centromeres of the bivalents, okay, line up, okay, and at the center, so that they are on opposite sides of the plane. So is it with this? And that is very important because with this orientation, the centromeres of the homologous chromosomes or homologous pair are pointing toward the opposite poles of the cell. So that this sets up in the anaphase 1 the separation of these homologous chromosomes, which have already recombined. So if you notice that there are already different segments because of recombination that happens in prophase 1, but it guarantees these orientations of the centromeres of the bivalent lining up on the opposite poles, guarantees during anaphase 1 that the homologous chromosomes are going to separate and move toward the opposite poles of the cell. And because of that, what we have is therefore a complete segregation of all bivalents, okay? And the consequence then is that when telophase happens, and with subsequent cytokinesis, the daughter cells are now possess half the number of chromosomes. Because if you now count the number of chromosomes in each daughter cell, If you, we all started with, if you count the centromeres here, in metaphase 1, you have, by counting the centromeres, you have 1, 2, 3, 4. Whereas in after telophase, that and cytokinesis, you now have for each daughter cell 1 and 2. So you end up with 2, and this is diploid, and this is haploid. Okay, and in telophase 1, just like in mitosis, you simply reform the nuclear membrane. Okay, so you end up at the end of meiosis 1, you end up with 2 daughter cells that are haploid. Okay, so try to review that, very important. Again, this is will be evident with what the significance this is in Mendelian genetics when you go to chapter 15, because this explains the phenomenon of what we call as complete segregation as principle number 1 in Mendelian genetics. So the next stage in meiosis is meiosis II. And meiosis II is essentially equivalent in terms of the behavior of the chromosomes, of the individual chromosomes, like mitosis. Okay, so there's no need for me to belabor the stages here for interest of time. You can just go through each of these beautifully illustrated and labeled steps, but this is essentially very identical to the mechanics of mitosis, except that the cells, there is no DNA synthesis between the two meiotic divisions. So there is no DNA replication that is happening at this point. Okay, so you actually start with a haploid cell dividing. So that's the only thing that is different. Okay, because there is no DNA synthesis. So in meiosis II, as I say here, the process resembles that of mitosis, except that the nuclei in prophase II have the haploid number of chromosomes, but not the diploid number. So at the end, at telophase 2, you end up with 4 daughter cells. And if you notice, each of the 4 daughter cells have their own centromeres, and they are made up of single strand, not double strand chromosomes. Okay, so please take note of that. So let's just go over briefly into the steps of meiosis 2. In prophase 2, the nuclear envelope again breaks down, and the chromosomes once again condense. And in prometaphase 2, the spindles again would attach to the kinetochore in the chromosomes, and in metaphase 2, chromosomes will again align in the center of the cell, individual chromosomes, just like in mitosis. Then in anaphase 2, the sister chromatids will now separate, having their own separate centromeres, because the centromeres also is going to split at this point, again, just like in mitosis. And then in telophase 2 and subsequent cytokinesis, the nuclear envelope reforms and the cytoplasm divides. So meiosis 2 is therefore considered, because it is mechanistically identical to mitosis, they are called the equational division, because cells in meiosis 2 have the same number of chromosomes at the beginning and at the end of the process. Whereas meiosis 1 is called the reductional phase, because you start with a diploid or a 2N and you end up with an N. So overall in meiosis, you end up with 4 cells that are haploid. Okay. But this is called equational because you start with an N and you end up with N. The only difference is that the N here is made up of sister chromatids per chromosome. Okay, whereas here at the end you end up with a chromosome that is single-stranded. So the sister chromatids acquire their own independent centromere. But you also notice how the chromosomes look like. At the end, you end up with 4 end cells that are unique to each other because of the crossing over that has taken place in prophase I of meiosis. So if you notice, I keep repeating certain elements here because those are the ones that are very critical for you to remember and understand. So let me summarize some of the things that I already mentioned when I was explaining meiosis in comparison with mitosis. So first, to highlight here, and figure 11.12 is an important figure for you guys to go over and read more carefully in your textbook. I would just highlight the key points for the interest of brevity. During meiosis I, the maternal and paternal homologs separate from each other. Okay, whereas in mitosis in mitosis, I'm sorry, the maternal and paternal homologs, they are all just aligned on the same plates. During meiosis II, sister chromatids are the ones separating from each other, very similar to mitosis. 3, the similarity of meiosis, if you look here, okay, there is some mechanical similarities in all of the stages, but most strikingly, meiosis II and mitosis is very much identical to each other. So the similarity of meiosis II and mitosis suggests that meiosis likely evolved from mitosis. The idea is that the mitosis method of division occurs in all eukaryotes, which is evident, that is a fact, and was certainly present, therefore, in the common ancestor of all living eukaryotes. If we assume that all organisms start from the same ancestors. Meiosis is present in most, but not all eukaryotes. And therefore, because the steps of meiosis are the same in all eukaryotes, meiosis is thought to have evolved in the common ancestor of all eukaryotes and has been subsequently lost in some groups. So think about this interesting pattern carefully. So this is this comparison provides a clue as to how these two processes have evolved. So let's try to compare the meiotic cell division in females and in males using the humans as an example. The cytoplasmic division in males and females are starkly different during the process of meiotic cell division. Okay, so they differ from each other. First off, if you compare those critical stages of the process of meiotic cell division, in both female and male, before meiotic cell division happens, the DNA, of course, replicates. That's identical. They go through essentially the same mechanisms of meiosis I, the first meiotic division, and then the second meiotic division. However, the daughter cells that are generated are quite different in that in females, here, if you notice, most of the cytoplasm is retained only in one of the meiotic product called the oocyte, which can develop into the functional egg. Okay, the other products would receive only small amounts of cytoplasm, and these smaller cells are called polar bodies. In males, However, the cytoplasm divides fairly equally, resulting in products that all form functional sperms, okay, by a process called differentiation. But during sperm development, most of the cytoplasm gets eliminated, and what remains essentially is the nucleus in the sperm head and a nice uh, tail-like structure called the flagellum, which propels the sperm toward the egg in order to um, allow fertilization to happen. And so, just to highlight the consequence of meiosis in the females and males, the products of meiosis in both sexes are called gametes. We have encountered this term before. Gametes are simply what we call as sex cells. Each gamete is haploid, containing a single set of chromosomes, and during fertilization, gametes fuse to form a single cell called, a new term, a zygote. This process therefore restores the original diploid chromosome number. So this explains the establishment and maintenance of a diploid state in sexually reproducing organisms because of the interplay between meiosis and fertilization. So this ends the lecture on 11.3. It's important for us to make sure to answer the RIF questions in the next two slides. So the first question is, homologous chromosomes are A, chromosomes that are similar in their size, shape, and gene content, or B, similar chromosomes that are found in different individuals of different species, or C, the two chromatids in a replicated chromosome, or D, any chromosome in a diploid cell. So please open your RIF account and answer this question and the next one after this. So the RIF 2 question is, crossing over of bivalents in meiosis occur in A, Prophase II, B, Prophase I, C, Metaphase II, or D, Anaphase I. Okay, so that concludes this video. I hope to see you in the next segment, section 11.4. Thank you.